Great. We've got about 5 or 6 minutes, so we've got a time for maybe one or two questions before lunch. Yeah, right. We've heard nutrition and now food safety, and now we'll go to lunch, so it'll be great. Let's start with sort of a basic science question here. How are pathogens actually transferred to the edible portions of a crop, and can pathogens move from soil into the plant and through the plant? And how do they get to the place where we would consume them from the soil? Everybody nods, but nobody's taking the mic.
Okay. So is this microphone on?
Is it?
Testing. Oh, you turned it off. Dr. Milner turned off my microphone. So, all right, the mic got handed to me. So this goes back to my postdoc years. I started this wonderful project. This is the beginning of my postdoc under the Food Safety Lab at Agricultural Research Service under the tutelage and direction of Dr. Manan Sharma. So he gave me a little task, and this was stemming from the 2006 baby spinach outbreak in Salinas. There were a lot of questions around that outbreak, one of which was, can human pathogens, namely O157H7, infiltrate through the root systems of baby spinach, go internalize into the tissues up to the edible portion of the crop that can be harvested? This is a serious issue because if you harvest and wash the baby spinach, the internalized pathogens you're not touching them. So this was a big concern. This was I crafted an entire postdoc career based on this question. I came at it from multiple different angles, and I can de facto, and I know there's still miscommunications and misconceptions around this issue, so I can de facto tell you that under normal agronomic conditions in the field, even using raw manure as a fertilizer, which you cannot do under LGMA restrictions currently, but if you do and you plant baby spinach to amend your soils that is contaminated with O157H7, the chances of those pathogens going into the edible portion of the tissue is almost nil. We're not going to see this happening under normal conditions. Now let's switch. Is it possible? Yes, of course it's possible. I've seen this myself under extreme conditions where basically in the laboratory conditions on a bench, you have to stack the deck to prove a theory. Yes, it is possible for an E. coli to go through the damaged root tissues up into the edible portion if you force them to do it. You can do that. This is more likely to happen under hydroponic conditions. If you have a soup of E. coli and you're growing baby spinach from seeds in that soup, you're most likely going to achieve contaminated product. And this is the same thing we see in sprouts and sometimes microgreens as well. So, but in the soil, root to fruit contamination in the soil is unlikely. FDA is doing some very excellent experiments to understand this further. We have seen fruit internalization of pathogens when flowers are contaminated. If you contaminate the flower of a tomato crop, 70% of those fruits that are developed from that flower will have internalized salmonella, and we've seen the same with cucumbers. We're expanding those research studies to other commodities to see how deep that rabbit hole goes. But from root to fruit, I'm not worried about that one.
All right, very good. Let's.
I was just going to mention briefly that with the viruses, we've actually seen the same thing. We looked at 2 different studies, one with soil and hydroponic. Hydroponic, you get a high risk, of course, because the viruses can flow throughout the plant. But even with root damage, it's more likely that the viruses are interacting with the soils than going up into the plant.
I want to direct the next question to you, and this will be the last question. So, and I don't know that the premise of this is laid out, and then you can respond. It says the waste treatment plant in DC is selling treated biosolids. So you nod, you must know about this. Okay. Which apparently are being analyzed for how safe they are. But the question is what's our level of confidence regarding viruses that you talked about when we put these on farms and garden soils?
So we have looked at some of these Class B or Class 2 biosolids that are supposedly treated and validated. As long as there's a validated treatment, it's heat treated, they should be fully inactivated, no viruses that are viable that are present there. The studies that we've done, we have inoculated with viruses, so I'll just clarify that. Although I will share with you that we're doing research now where we're studying reclaimed water that comes from treatment facilities, and we do find protozoa and viruses in the water samples. But I'm going to share this with Pat because I think she's also done quite a bit of work with this, but it's my understanding that the validated process is enough to inactivate those.
Yeah, heat treatment is, when done properly and under exacting controls, it's going to kill all these different agents. The specific example that you asked about with the DC situation is they have a whole new treatment system there, in which case it's actually a two-step system. It's a thermohydrolysis system. Basically, they autoclave the solids first, and then after they autoclave them, they put them through a digester. So the product that's coming out now from DC water is actually a thermohydrolyzed digested material. And we've tried recently to re-inoculate that material to see if we can get E. coli to grow, and we have so far not been able to do that.
So it killed future pathogens. Huh? Yeah, go ahead.
I just wanted to make one comment, and this wasn't based on a question or anything, but something I wanted to add. So there was a question of antibiotic-resistant pathogens and potential hormones and other chemicals coming off of feedlot manure. And I can tell you that there's one wonderful way of eliminating all these residues from before you add them to your soils, and that is composting. Composting, and Dr. Milner has done a lot of this research to show that proper composting eliminates the antibiotic resistance pathogens and eliminates the antibiotic residues, and it eliminates hormones. So,
Well, I'm glad you made that comment because that was a question that I had on my list, but we've run out of time, so you took care of it.